All right, we are live. Welcome, everyone. Today I am with Dr. Dan, aka CreationMyths, and we're here to talk about vaccinations, virology, and all sorts of nerdy stuff. So, Dr. Dan, welcome. Please feel free to introduce yourself.
Hi, I'm Dan. I'm an evolutionary biologist. My specialty is viruses, and my channel is called CreationMyths, where we we treat creation science with exactly as much seriousness as it deserves. So we actually
zero.
That is to say, we'll start by saying, let's take this seriously as a scientific idea, and then we'll look at very specific claims and examine them and see if they actually stand up to scrutiny. And so far, we are 0 for about 19.
Okay, and how did you? Yep. And how did you get started with responding to Young Earth creationists, and why do you feel that it is important to respond to their claims?
So, and how do you feel what?
Why do you feel that it is important to respond to their claims?
Well, so the how I got into this is a really funny kind of sideways thing. How much time do you want to spend on it? Because I'll give you the less detailed version, and then we can talk more about it at some point now or not if you want. But I had to take a class my first semester of college that you had to take a class. It was part of this thing called the freshman seminar program, and you had to take a class that was outside of your major. And I picked a philosophy class called "Do God and Science Contradict?" That I thought that sounded interesting. Sounds interesting. Let's take this class. Turns out it was taught by a creationist who taught it as a pro-intelligence design propaganda class. So that was really fascinating. And it became like a weekly fight against the misinformation in that class. It became really confrontational, actually, between me and a couple of the other students and the professor. I was a bio major, my roommate was pre-med, and there were a bunch of other biology people in that class. And it was like a weekly, we would get together and figure out how we were going to fight back against the stuff that she was lying to us about. And I learned so much about evolutionary biology that semester. It was that class that got me not just really into the evolution versus creationism thing, but also got me really into evolutionary biology. It's because of that class and that awful professor that I got into evolutionary biology. And why does that matter? Why does it matter that we deal with young Earth creationism? It's a fair question that people could be skeptical about, right? Because what harm does it do if people think the world is 6,000 years old, right? Okay, who cares? That doesn't hurt anybody, right? Wrong. Because what you're doing, if you can train your brain into believing something as obviously false as the idea that the world is 6,000 years old, then you can train your brain into believing other obviously false things that do have real world consequences. So you're basically practicing tricking yourself and convincing yourself to believe something that's not true, and it makes it easier to buy into all sorts of medical woo, to anti-vax conspiracies. It makes you more susceptible to all this other stuff. And we see a correlation between young Earth beliefs and a bunch of these other things that are tangible problems in the world, rather than just a wrong thing that you believe, right? So that's why it's not the kind of claims that matter kind of for their own sake necessarily. It's the we shouldn't indulge people that are teaching other people how to believe wrong things, because that is bad applied globally.
Okay. So let's get into some topic about vaccinations, which is one of my personal favorite subjects. What exactly does it mean for a vaccine to be, say, 95% efficient, since there is a lot of confusion about these numbers?
Yeah. So before we even do that, let's just real quick what a vaccine is, right? Because a lot of people aren't even clear about that. So a vaccine is just something that you introduce into your body that stimulates an immune response. And then the vaccine itself doesn't make you sick. It doesn't give you any kind of disease or anything, but it triggers an immune response to a specific pathogen. So then the next time your body sees that pathogen, you actually can mount a stronger response to it. You've practiced, right, against the vaccine. So now when you see it for real, you can really fight back and you don't get sick. It prevents you from getting sick. And there's a huge range of efficacies for vaccines. And when you read that a vaccine is 80% effective or 95% effective or whatever the number is, all that means is that that vaccine provides a 95% reduction in risk of getting that disease, right? It's not saying that 5% of people that get vaccinated will get the disease. It's not saying there's a 5% chance of getting the disease. It's saying whatever the risk is, if you are not vaccinated against the disease, you are reducing that risk by 95% if you are vaccinated and the vaccine is 95% effective, right? That's
Right.
what we mean when we say efficacy of a vaccine.
Right. So if a virus would say you have a 1% chance of getting vaccinated, of getting the disease, and you have a 95% chance of getting vaccinated, your chances of getting the disease would probably be like 0.001% or something like that.
Yeah, so I always think of it in fractions because it's just easier in my head. But if you have like 1 out of 100, right? If it's if you do something, some activity, right, and it's a 1 out of 100 chance of just baseline getting infected by whatever it is, and you have a 95% effective vaccine against that thing, well, that's a reduction to 1/20th, right? Because 5%, right? So you're going from 100% susceptible, like whatever that baseline is, right? It's arbitrary, and then reducing that by 95%. So you're reducing it to 1/20th of the original risk. So if your baseline risk is 1% or 1 out of 100, your new risk is going to be 1 out of 100, 1 over 100 times 1 over 20, right? So now you're taking your 1 out of 100 risk and you're reducing it to 1 out of 2,000. And that's obviously a big difference. That's a lot less risk.
Right, of course. And I really wish there was more information about that, since there is so much confusion. So what are the different types of vaccinations out there? Because I know Moderna and Pfizer, I believe, are both mRNA vaccines, and I'm not quite sure which one the Johnson & Johnson. And I must say that I'm really fascinated with the whole mRNA vaccinations.
Oh, they're so cool. They're so cool. So let me just take 1 second, and if it's okay, I will pull up a slide that I made for a kind of a local. I did couple months ago. I did a little presentation for my town for a group in my town that was doing vaccine advocacy stuff, and I volunteered to participate as kind of like I'm going to tell you about vaccines person. And so if you don't mind, I'll just share the slide that I made for that has the different types of vaccines, and we can talk about the different types. So here we go. You should see it down there. There it is. So this angry guy over here, that's obviously the virus, right? And then I'll just tell you the spikes sticking out of it. Those are spike proteins, right? Now every virus is different in terms of its structure, right? So it's not spike proteins for every virus, but like, it's an approximate. We're dealing with coronavirus, right? So we talk about spike proteins. So one way you can vaccinate against something is you make what's called an attenuated vaccine. This is the oldest way of doing vaccination. So this is where you take the actual pathogen and you grow it usually on a different host. So like, for example, for polio, we used an attenuated vaccine for a long time by taking the pathogenic virus and by having it infect monkey cells, or I think it's monkey cells, or maybe chimp. It's some other ape, and or not ape, some other primate. And it adapts to the other host, and then it becomes bad at infecting its regular host, which is humans. So you're exposed to the weakened form of the virus, the attenuated virus, and then your immune system can respond to it, and it can still infect your cells, but it's slower, it's weaker, it's not as good at infecting you, so it can't make you sick. So then you respond to that weakened virus, and then when you see the real one, you can fight it. Then we have the inactivated vaccine. So this is where you just take your pathogen, you kill it with like heat or chemicals in some cases, so it's dead, it can't infect you, and then you inject like the whole virus, and your immune system sees it and goes, that thing's a virus. I see that virus. Doesn't matter if it's not doing anything. It's still a virus. And then so your immune system makes the antibodies and you learn how to deal with it. And then same thing, you're good next time. Those are both pretty old types of vaccines. The more recent ones we have are the subunit vaccines. So this is where instead of taking the whole pathogen that can make you sick, you take just a little piece of it. For example, if you were doing this for coronaviruses, you might take the spike proteins, because that's the one that our immune system sees, is the spike proteins, and that's what we make antibodies to. So you take the spike proteins, these little red spikes, right? And you isolate those, you make a lot of those, and then you inject the spike proteins. And it doesn't matter that the rest of the virus is missing. Your immune system still sees those spike proteins and goes, aha, virus. And then you do the same thing, you respond to it, you make antibodies, and now you're good. So those were the types of vaccines we were using as of a couple of years ago. Now we have two more that have also entered the toolkit. So one of them is slightly older. These are both very new technologies in terms of their clinical use, but one of them has been in development for a little bit longer. Those are the viral vector vaccines. And this is the J&J and AstraZeneca coronavirus vaccines, or viral vectors. So what this vaccine involves is you are injected with a different virus, but instead of that virus containing its own DNA, it contains the DNA to make the spike proteins for whatever you want to vaccinate against, right? So in this case, the SARS coronavirus 2. So you inject this virus, your cells, that virus gets into your cells, and then your cells will express the DNA in that viral coat right in there. So that's your DNA in there in blue. And when your cells express the genes on there, it's not the genes for the virus you're using as a vector. It's the genes for the spike proteins for the coronavirus. So then your cells, your own cells, make a bunch of the spike proteins, and then your immune system goes, what are you doing? That's a virus, kill it. And then you make the antibodies and you deal with it. And now you're good. And then the last one, the newest kind, and this is what we see for Pfizer and Moderna, are the mRNA vaccines. These are so cool. These are so darn cool. What this is it's a little bit of RNA, or if you want to be more specific, it's the mRNA, the messenger RNA. So the things that your ribosomes see and will make proteins, they'll do translation to make proteins off of. And so you inject, this vaccine. It contains the mRNA. The mRNA gets into your cells. The mechanism doesn't matter. It gets into your cells. And then your ribosomes express that mRNA, churn out a whole lot of spike protein in this case, right? You just generate tons and tons of spike protein. And then your immune system sees all those spike proteins and kind of freaks out a little bit. And that's what it's supposed to do, because now it's on high alert and it knows how to deal with the threat. So when someone with COVID coughs on you, your immune system can handle it. And that's how it works. It's really effective. It's really cool. So those are the 5 types of vaccines that we have in use right now. And specifically for coronavirus, we have the bottom 3 are all being used. There's a bunch of different companies developing subunit vaccines, which are simpler to make and cheaper to make than either vector vaccines or mRNA vaccines. So if you want to immunize 7 billion people as fast as possible, great that we have these new technologies, but you know what? Subunit vaccines work darn good too, and we want to churn those out as fast and as easily as we can. So we're using all 3 of these actually for coronavirus.
All right, awesome. And what type of viruses are there, and what type of virus is the COVID-19 SARS-CoV-2 virus?
Man, there are so many types of viruses. That's a fun thing. If you want your head to hurt, is look up viral phylogenetics, because it is a mess. Here, I'm going to take down the, I'll take down the, here we go. We can stop the screen now. So there are so many types of viruses. And the fun thing about viruses is they're polyphyletic. So they're not all related to each other. They don't have universal common ancestry like cellular life does. So you have all these different types of viruses that operate very differently. They look very differently. There's no homologous genes between the different groups. So we don't even have groups like kingdoms or phyla for viruses, because you can't get common ancestry in terms of broad enough groups to have those things. So you have species and genera, you have families and orders. And off the top of my head, I should know if you go above order, do you have, I don't even think you have classes. I think it's just orders. And it doesn't go any higher than that, but don't quote me on that. Maybe there's classes, but I think it's just orders. And so above that, it's based on their genomic architecture. So that what we mean by that is, what does their genome actually look like? Like, what is the physical structure of its genome? And for us, for all cellular life, our genomic architecture is double-stranded DNA, right? That's it. That's our genomic architecture. Well, viruses can be double-stranded DNA. They can be single-stranded DNA. They could be double-stranded RNA. They could be positive sense single-stranded RNA. They could be negative sense single-stranded RNA. And then they can be retrotranscribing. So that's like HIV, like the retroviruses. So that's where they do this funky thing where they start as RNA, but then they have a DNA intermediate, and then they do their genes off of that. It's weird. And then there's another one that's similar to retrotranscribing, but it's got some quirks. I forget specifically what those are called, but I think the hepatinoviruses are an example of that, if anyone's curious. But those are the different types. And then SARS coronavirus 2 is a single-stranded RNA virus, and for the life of me, I don't remember if it's positive sense or negative sense. So let me see. Hang on, I'm going to look it up right now because I don't remember.
Yeah, it looks like coronavirus, highly positive sense single-stranded RNA virus. Like
positive sense. There you go.
So what does it mean for it to be, say, positive sense or negative sense?
Oh, yeah, we're really getting into the weeds. This is good. So positive versus negative sense for RNA refers to whether it's the coding sense or the amino acid encoding orientation for the RNA, or if it's the complement of the amino acid encoding sense. So if you have RNA that is positive sense with the right end pieces, the 5' cap and the 3' tail, that positive sense RNA, that is mRNA, which means you can express proteins directly off of it. You could just make proteins directly from that thing. If it's negative sense, that means that it is the complement to that. So think about like your DNA and your double helix. The two strands are complementary to each other, where A always pairs with T and C always pairs with G. RNA works the same way, except it's U instead of T, but the pairing works the same. So a negative sense single-stranded RNA genome is not in the coding orientation, so you have to make a copy of it, right? That's then in the positive sense, and then that copy, that's your mRNA that you're going to use to actually express your genes. So so these viruses, the SARS coronavirus 2, is positive sense, which means its genome itself, when it gets into the cells, can just be directly read as mRNA. Super cool.
All right, so there has been quite a bit of controversy recently about the origins of SARS-CoV-2, and some news articles about possibly being a Wuhan accident or leak from the Wuhan virology lab. I'm not sure if you have seen any of those articles, but how credible is the Wuhan lab leak hypothesis, and how seriously should we take it?
So the thing here that I want to emphasize before we get into any of the nuts and bolts of this is that the media has been all over the place with this one, just totally boomerang of like, it's definitely zoonotic versus it's definitely from the lab, and back and forth with every story you get like, oh, the consensus is shifting. And that's not really reflective of where the science has been. The state of the science right now, and I don't want to get out ahead of my own expertise in this because I have not read all of the papers in this. I've looked at a couple of different things, some phylogenetic stuff, looking at this genome, and then I read what people who are doing this work day to day are saying. And what I'm seeing from those people is that not just right now, but from the beginning, basically, the consensus most likely source was a zoonotic transmission, so a transmission from some other animal into humans, with a non-zero chance of it being from a lab. And then with the caveat that if it's from the lab, it was probably something that was isolated from nature and stored in that lab, and not like some bioengineered, possible like bioweapon thing, right? And what people have been saying the whole time is look into everything, get as much data as you can, look into everything. Don't stop looking at the lab hypothesis. Don't stop looking at the zoonosis hypothesis. Just get the data you can and look into everything and figure it out. And as it's been going on, the consensus, not like a 99-1 consensus, but one person I read said it's 75-25 or 80-20 consensus, is that it's zoonotic. Now, that's one person's opinion, and honestly, I'd have to go back through and find out who it was. I have to find the piece I read where find the person who they quoted. But, you know, there's one person's opinion on kind of the state of where it is. But based on what I'm reading and the different sources and the frequency, that seems like about right to me. And looking at the phylogenetics, if you look at a phylogeny of the genome of SARS coronavirus 2, I mean, it falls out in the middle of a clade of bat coronaviruses. It's bat coronavirus, bat coronavirus, bat coronavirus, SARS coronavirus 2, bat, 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 bat. And then actually on a not that far branch is SARS coronavirus 1 over here too, also surrounded by bat coronaviruses. So it seems like, you know, it sure seems like it's a zoonotic transmission. Is it possible that we get new data at some point in the future that makes that less likely to be the case? Absolutely. But right now, as far as what we know and as far as what I'm familiar with, what I've read, it seems like a zoonotic transmission is the most likely explanation.
Right. And there was a lab hypothesis. It may have been something that was in the lab, but it accidentally got spilled out and all of that. And that's how everything started.
It could, yeah. I mean, it could be as simple as, you know, people try, if it was, what it would be is because you have a virus lab, what you're going to do is a lot of that is isolating stuff from nature. And then, like, if you got to transfer stuff, if you're testing out viruses on different model organisms and things, like, you can get sloppy and get infected with a virus in the lab, and then you take it home. That can happen. I mean, that happened with when they discovered the Mrna virus, one of those giant viruses before they realized it was a virus. Turns out those things can cause pneumonia. They figured that out because a handful of the people working on the initial characterization got pneumonia, and they were like, what in the world kind of pneumonia is this? And it turned out it was those giant viruses they were actually working with in the lab. So if you don't know what measures are reasonable for the thing you're working with, or you cut some corners with the protective measures, then it's possible you could have an infection in the lab, and then it's going to get, I mean, we talk about like lab leak, and it brings to mind like ideas of like there's like a glass vial somewhere that cracked and there's like virus literally leaking out. That's not what a lab leak looks like. Like a lab leak is where it infects somebody in the lab and then that person goes out and spreads it outside of the lab, right? That's what it would look like. And if they have just a strain stored in the lab that was isolated, you know, from bats, right? Then like, where'd it come from? Well, it came from bats. So like the question of did it come from the lab or not is a different question from what was the reservoir in nature that it came from, right? Those are two different questions, and they're not and the answers are not mutually exclusive necessarily, right? So that's a lot of the reporting is conflating that and saying it has to be one or the other when they're not necessarily mutually exclusive explanations.
Right. And this is one of the reasons why I absolutely hate the media when it comes to science, is that they are, I think, deliberately sensationalizing things and spreading disinformation that ultimately harms public science and public health because they're not doing their job properly and they're just going to be doing whatever makes the most money.
I think it comes down to exactly what you said. There's a lot of there's two things. One is the bias towards sensationalism, and then the other thing is that at most publications now you don't have like a dedicated science reporting staff anymore. So in 20, 30, 40 years ago, you might have somebody on staff at all your major newspapers and all your major networks that is like the science person that would like, it's their job not just to like know kind of the ins and outs of the science better than most average reporters where it's not their field, but also to be able to read scientific publications more effectively than somebody else, right? That's not trained to do that. And most reporting organizations now, that doesn't exist anymore, right? So you have a lack of expertise on the people that are kind of the middlemen and transmitting the message, combined with a bias towards being sensationalist and a bias towards conflict and novelty that leads to every report where you get a new paper that shows that, well, it's probably a zoonotic transmission from whatever population of bats. Oh, we've confirmed whatever. And then 2 weeks later, oh, new evidence. It's almost certainly a lab leak. Like, No, we're not talking fluctuations of 90-10 probability in one direction back to 90-10 in the other direction. We're talking fluctuations from 75-25 to 80-20 and back, right? We're talking very small changes in like the actual state of where the consensus is being reported as these giant reversals. And it's very frustrating to read this. Very frustrating.
Oh yeah, definitely. And I think there definitely needs to be more accountability within the media and more accountability for when people blatantly make these types of mistakes. Very few people, very few reporters are even willing to acknowledge that they made a mistake.
You get like stealth corrections on things without like an official acknowledgement of corrections, or you get a correction, you get a front page thing of like new evidence of lab leak, and then a week later, you know, that'll be like the New York Times like Sunday headline on the front page or something, right? And then, you know, 9 days later on page 10 of the Tuesday edition, right, it'll be, you know, new evidence, blah, 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 and it'll be kind of walking that back a little bit, but like how many people are going to read that first headline versus the whole second article? So you end up completely messing with the understanding, the general understanding, and kind of doing the opposite of what the media is supposed to do, which is inform people accurately. It's like, no,
Right.
You're doing the opposite of that, and you're misleading people. But here we are.
Yep. All right. All right. So, are you doing any research at Rutgers, or are you just doing any, or are you just professor? What type of
Full-time
research are you?
teaching. I am full-time teaching, and it's fantastic. I super burned out on research after grad school / during grad school. Super burned out. So getting a break from that. I don't know if I'm ever going to go back to it. Right now, my position is not, and that is fine with me because I really dig the teaching side of things. That's super fun. And had no idea that was going to be the case, but it turned out that was the case, and it worked out really well. So I am full-time teaching, and I am thrilled about that. I don't have to worry about grant money. I don't have to worry about it. It makes me very happy. One less thing to worry about.
I don't have to worry about writing a whole long paper just for it to be rejected or having to pay a $3,000 publication fee to whatever journal it is.
Yeah, it's and you know, not all the journals where the authors have to pay are bad. I mean, a lot of open source journals where you know, a lot of journals will get their money through subscription fees, which I think is wrong, predatory, especially since most of the science is funded by the federal government, and it's like, well, it's publicly funded, so like the research should be publicly available, but you know, it's not how it works.
And
that's not how it works.
Right, and some companies, I'm not going to say their names, charge millions of dollars to universities just to have access to their journals.
Elsevier, screw them.
Yeah,
I'll say their names. Elsevier.
Yeah,
and who else? I forget the other big ones. Elsevier's they're terrible. I guess they're awful.
Yeah, they're a terrible company, but I know they have pretty solid journals.
Yeah, I mean that's the problem, right? It's not quite a monopoly, but it's close. So it's like a triopoly. So it's like, what are you just gonna like not read any of the things? Like, no, you need to get it. You need to have access to it. So what are you gonna do? You know, it'd be great if there was like a hub online where you could get your science. That would be really convenient for everybody. But yeah, if there was a science hub of some kind where you could access stuff.
Yeah, there actually is. They made a Sci Hub. Which is
that's what I was alluding to without saying.
Yeah.
I don't want to say it, but you know.
Yeah, one of my favorite.
I use their
one of my favorite things.
Sorry, what are your favorite things?
I can't remember what we were talking about. So I guess we're going to go into evolutionary biology, and how are phylogenies constructed. So we were talking about how phylogenies are, of course, one of my personal favorite things in biology. So so when we take these SARS-CoV-2 phylogenies, how exactly are they constructed?
Well, so it is a whole process, and the way people learn how to do it is not the way it's done anymore. So I'll start with the way you like learn how to do it, like the basic way, where you take your nowadays it's all going to be molecular data. It's all like DNA sequences and sometimes amino acid sequences. So what you do is you take your DNA and you line it up so that all your different taxa, all the different groups you wanna do your analysis with, they all match, right? They all line up. So all the parts that are the same are all lined up with each other. It's called an alignment. So you're going to do an alignment first, and you'll have, maybe, I mean, I'm familiar with this with viruses, and you'll take like 100 or so viruses, and you'll have, a couple thousand bases that you'll line up their genomes. You do an alignment. And then what you do is you put your alignment into a program, because like we do not have the computational power to do this by hand anymore, right? So you have to have some computer program that does this. And what you do is you give the computer program constraints. You give it parameters. You say, okay, so mutations going from C to T are more likely than going from A to T, for example. So if you see a number of if you see a C to T change at the same place in two different like sequences, you'll count that as less likely to be due to common ancestry than if you see a different mutation that's less common. You have to have ways of accounting for gaps in your sequence due to deletions or duplications. So you have to, it's complicated math to kind of tell the program, like, what are the parameters you're dealing with? You're also telling it things like population size. So you give it all these, parameters and say, here's how we think these evolutionary processes work. Here's all the sequences. You, computer program, tell me what the most likely arrangement is in terms of their common ancestry, right? So you're using these techniques called maximum likelihood techniques, where given the parameters, what's the most likely evolutionary history for all of these related sequences? And I want to be very clear, we're talking about the sequences because you can have differences between different genes within your group of organisms. You could look at a bunch of organisms and compare one gene and get one phylogeny. And then a different group, you compare a different gene, you get a different phylogeny for a number of different reasons. So you have your sequences, they're aligned. You have all the rules that govern their evolutionary processes. And the computer then does some kind of Bayesian process. So you're talking about Bayesian statistics here, where what you're doing is you are basically, it's forward-looking rather than backwards-looking is how I think about it. So you're, instead of saying, getting like a p-value, where you're saying like, what is the probability of this not happening, right? You're kind of going the other way around, and you're saying like, you're proposing an affirmative rather than the converse of what you're viewing. And so you construct a bunch of different phylogenies. At first, it's pretty close to random, right? And then the program evaluates them for their likelihood. And then you say, okay, let's take the best 4 or the best 10, and let's modify them and make another 10,000 phylogenies out of them. And then let's evaluate those, and we'll take the best 4 again. Okay, now we'll spit out 10,000 new ones. And you do this iteratively, like 1,000 times or 10,000 times. And you can imagine how computationally demanding this is, right? And then at the end of it, you start to converge where all your phylogenies start to look really similar to each other. And that set of phylogenies that you get at the end represents your most likely set of relationships, your most likely evolutionary history for the groups of organisms that or the groups of sequences that you're looking at. So you're using these very computationally demanding processes, given a set of parameters and a set of sequences, to propose what the most likely evolutionary history is. And I want to be very clear about this. When you make a phylogeny, what you are doing is you are making a hypothesis. And then what you're doing with subsequent phylogenetics work is like looking at other genes, looking or adding additional organisms to your phylogeny. What you're doing subsequently is testing that hypothesis. Does your phylogeny stay the same? Or does it change when you add more data or when you look at a different gene, right? So you're generating a hypothesis, and then you're going to test it. And at the end of the, and you're going to come up with a better hypothesis, and you're just iteratively improving your understanding every time you do it.
Right. And what's really cool is that you can do different part of the genomes and test the phylogenies and have like a cross check. My personal favorite, my personal favorite is endogenous retroviruses, and how we can use those and create a phylogeny based on the endogenous retroviruses and match that up with other parts of the genome.
Exactly.
Right.
So it ends up being, so you end up with the phylogeny at first becomes it's a hypothesis, right? Here's the expected relationship. But then you could say, okay, let's test that. Let's look at this other part of the genome, or let's look at these other things. Instead of this one particular gene, let's look at these endogenous retroviral insertions. And then you could do phylogenies on those other things. And then if you get something that matches, now you end up with evidence for your proposed evolutionary relationship, right? Now you've generated evidence in favor of your hypothesis. Oh, I see a bunch of messages just got nuked. What's going on there?
Oh, just you can see them in the streamer thing.
I can actually, I can.
Yeah, anti-LGBT stuff.
What's this?
So that is why they got banned. If you are promoting anti-LGBT,
yep, there you go.
You're going to get banned. Anti-vaxxers.
Yep, you get banned real fast. Yep.
And here's a question from CreationMyths. Do you consider different mutations types like Endel's duplications gene movements while constructing phylogenies?
You sure do. Yeah. So what you do is part of the parameters that you're using to build your phylogeny is called a substitution matrix. So you are basically scoring any differences that you find. So you have like, for example, you'll look at different types of point mutations, right? You look at A to T, A to C, A to G, C to G, C to T, right? You'll look at every possible point mutation and say, okay, how likely is that? Or what's the relative frequency? So we could score those in terms of how different we're going to count the sequences based on those. But you can also score insertions and deletions. And you can assign, depending on the way you're approaching it, you could assign different scores for those things. You could do the same thing for inversions, right? So if you have a region right here, and then in a different sequence, you have the same region, but the orientation is the other way. Well, how does that count, right? So that's one event, but then all the letters within that region stayed the same, right? So how do you score that? There's debate about the best way to score things like insertions and deletions, and different approaches will get you different kind of final results. And it's all basically based on the relative frequency of these different things, right? So certain situations, certain species, certain processes for replicating your genome make certain types of mutations more likely or less likely. And then you can consider that when you're establishing your parameters to say, well, given the way this thing replicates, we know that slippage is relatively common, so we're not going to penalize two sequences for having insertions and deletions between them, because we know those types of things are really common during this particular type of replication. So we're not going to worry about it, right? And in other cases, you say, oh, this type of DNA replication is highly robust against insertions and deletions. So if we see an insertion or a deletion, we know that that's like a big deal because that's relatively uncommon, and we can draw our conclusions accordingly. So yeah, we absolutely account for all that stuff. It's going to be very specific to whatever group of sequences you're looking at, right?
Yeah. And one of the things that I know we were talking about on, I think, Sal's channel is that certain proteins do not share a common ancestor. And so the obvious question that I have regarding that is, how exactly do two organisms that may share common ancestors have proteins that do not? How does that happen?
Yeah, de novo proteins. That's like de novo genes are a thing, and we've gotten really good at finding them just in the last decade or so. So what we know to be true is that you can have biochemical activity from just random sequences of RNA or amino acids. If you just like random polypeptides, the likelihood that that random sequence of like a few hundred amino acids has some biochemical activity within a eukaryotic cell is like pretty high. It's not 90%, but it's also not 0.00001%, right? It's reasonable. So what we find, and then the second thing that we know is that spurious transcription, so just making RNA where there's no gene, happens all the time, or where there's no regulatory function, or where there's no RNA genes, right? tRNAs or ribosomal RNAs. You've got lots of RNA that's being made just like all the time, because the regulation is not 100%, right? It's a leaky type of regulatory network. So you make lots of random RNAs. And the likelihood that some of those RNAs have some kind of biochemical activity in terms of associating with other things in the cell is reasonable. And we also know from experiments, like direct experiments, that acquiring a start codon, a stop codon, it's actually not that hard. And I've actually directly observed this by trying to make knockout strains of a particular virus, where what you do is you have your gene that you want to knock out, and the way you knock it out is you just hit the front end of it, where translation would start. You hit it with like 2 or 3 or 4 stop codons. You just mutate it like 5 or 6 or 10 different mutations, so you have like a bunch of stop codons like right away. So it starts translating and it immediately stops. And if it reads through the first one, there's like 2 more to stop it. And me and my colleagues were trying to make these knockout viruses, and the darn things overnight, in like 12 hours of replication, they'd have so many mutations, they would get rid of all those stop codons.
Interesting.
Because the ones that had the gene without the stop codons were more fit within that population. Because if you could make the gene yourself rather than having to get it from the exogenous source, that was a big benefit to you. So you had just so many mutations, and you ended up just turning this gene back on. And so what we know to look for to find these genes, these new genes that can appear, is you find where in a bunch of things you have RNA that is being transcribed. And then in one or very few lineages in that same clade, you have actual genes. And it's like, what happened is that RNA that was being expressed acquired start and stop codons, and now it's translated. Now it's a protein, and it goes off and does something.
Yep. So debunk. So again, comparing the ratio of different mutation types of 2 organisms, we can directly prove the common origin, because the difference between them match the relatively frequency of the mutation types.
I don't know if I would say directly prove, but it's certainly like a point in favor of common ancestry, right? And so if you have, for example, like the one that I'm most familiar with is C to T mutations due to a process called deamination in single-stranded DNA. Like, the fact that those are so common, like, messes with things like codon bias, for example, right? And we can see that signature across all the different single-stranded DNA viruses. Now, that's useful phylogenetically, because we can then put that into our phylogenies when we're doing our math to figure out the relationships. We now know that we have this biased mutation rate, and that makes things useful. But it could also be misleading, because we know that single-stranded DNA viruses, as a group, are not monophyletic. One group of single-stranded DNA viruses evolved from plasmids that stole the capsid protein from a different group of viruses. So if you actually do that, they only have 2 major genes, and if you do the phylogenies, you get 2 different phylogenies for the 2 different genes. One of them is within this other group of viruses. They all have the same kind of coat proteins. And then the other one is actually falls out, it's all plasmids on that phylogeny. And then right in there's this one group of viruses that use this, it's called the rep gene. And so in that case, having the similar mutational biases is actually misleading phylogenetically. So as you can imagine, this gets complicated really fast.
Yeah.
And that's what makes it fun.
Oh yeah, definitely. I love these fun, complicated stuff. So do you have a favorite piece of evidence for evolution?
Favorite single piece of evidence for evolution? I don't know about, it depends on what you're looking at, right? Like human chimp common ancestry, like the slam dunk is chromosome 2. It's just like, there it is. That's the one that pretty much does it. ERVs are amazing because they're errors. They're not functional. They're from viruses, and then the viruses became inactivated. So like, there's no reason functionally they should all be like, they should match between species, and that you can make a nested hierarchy between different species according to their relatedness by other metrics. And yet we can do just that. But my absolute favorite, if you want to go large scale, my absolute favorite single piece of evidence for universal common ancestry of cellular life is that we all use double-stranded DNA. Because you don't have to. There's nothing that says you must use double-stranded DNA as your genetic architecture or your genomic architecture. And we know this is the case because we have the opposite. We have a group of organisms that don't share common ancestry. Viruses, polyphyletic. we have all these different groups of viruses, dozens of unrelated groups of viruses that all use different genomic architecture. If cellular life was polyphyletic like viral life is, and I'm going to call it viral life, and come at me, viruses aren't living people. If cellular life was polyphyletic like viral life, there's no reason that everybody's going to have the same double-stranded DNA, specifically using thymine instead of uracil.
Yep.
Like, that is slam dunk, monophyletic, universal common ancestry.
Okay,
Take it to
so
the bank.
some more here. So like the ratio of N-stel's 2-point mutations, 1/12.5 for great apes. So if we compare the genome of chimps and humans, we find 25 more single-point mutation difference. And N-stel's is true. Common designer could also create double-stranded DNA.
Oh yeah, I mean, a common designer could of course have created all the different cellular forms of life with double-stranded DNA genomes. Absolutely, but like that's just a unfalsifiable ad hoc. Like, yeah, of course it could have. Yes, it could have happened that way, but there's no way to test that. So, not a reasonable scientific hypothesis.
Right.
Also, there are tests we can do in other ways, right? So, for example, if we say that you have independent creation of different lineages, right? But they were designed similar, right? So, for example, humans and all the other apes were created separately, but they were designed to be very similar. And that's why the genomes all look very similar. And that's why you get this nested hierarchical pattern. It's an artifact of this common design. Well, you can make predictions based on that because there are parts of the genome that everybody agrees can vary randomly that don't have any effect on function or fitness or anything, right? Things like many synonymous codons or obviously non-functional sites like pseudogenes. I would also put ERVs in that category, but now you get creationists arguing that ERVs are actually mostly functional, which is clearly not the case. Or I've even had some people argue that ERVs are where viruses came from, so it went the other way, which is not a reasonable position to take. But so what you can do is you can look at these non-functional sites and you should see random variation and no nested hierarchical pattern in things like codon bias, for example, among great apes, or endogenous retroviral insertions among primates, right? Because they're non-functional, they are probabilistic rather than deterministic. So if they were created one way and then you have these probabilistic processes operating since creation, you should not see the nested hierarchical pattern that we see with the functional sequences. But what do we see? The nested hierarchical pattern, which like that is a direct contradiction of the design, common design hypothesis, right? Because then, okay, explain the non-functional parts. Well, you can't.
Yep. Answers to Atheism says Dr. James Tour just came out on Neil PP's channel as an evolution denier. So why does that not surprise me?
I thought he was already an evolution denier. I thought he was already like special creation all the way. I thought that was common knowledge.
Right. Yeah. And Tommy Emo. Common designer can explain any design because there's no rules applied. Pretty much that's the game.
It's not. It's depending on how you phrase it, it's either not a falsifiable hypothesis, or you're saying, well, no, you can test it. Okay, the test would be the non-functional parts of the genome, and then it's wrong. Then the data contradict the hypothesis. So it's it's squishy that way.
Yep. And Rio debunk asks, can we find out where the different virus codings came from?
Like the different genomic architectures? That's a fun question. And I have thoughts on it, but I don't have any evidence for it because we're talking, 4 billion years ago. And it relates to the metabolism first school of thought, or the processes first school of thought for abiogenesis, rather than the cells first, or the compartment first kind of approach to abiogenesis. And if you have processes first, right? Some of the processes you're going to have are going to be DNA to RNA back and forth, and DNA to RNA to protein, and RNA back to DNA, and all those kind of genetic processes. And I think that the way the different viral groups came about is you have different parts of these processes compartmentalized in different ways. And David, if you don't mind, I'll just take a minute to kind of like throw everyone's brain. I'm just going to warp how we think about viruses a little bit, just to give you an idea of what I mean. So
All right,
there's one large group. There's a large group of viruses, and the way they operate, and it's a large group, but it's also a group of large viruses. It's the giant viruses that are really fascinating. What many of them do when they get into a host cell is they wreck everything, and they rebuild basically a cell within a cell called virus factory. So it would be like the Mimi virus factory, for example. And so you've got like an amoeba that's infected by this virus, and inside the amoeba is essentially a smaller cell, and all that cell does is churn out viruses, right? Now, you could look at that as a virus that remodels the inside of a cell and then bursts out and transmits. You could also look at that as a prokaryotic endoparasite of a eukaryotic cell that transmits via a protein spore, if you will, with its DNA on the inside. So like that boundary line in situations like that between viruses and cells is a lot less clear cut than we often think of it. So when we talk about like, where do the different types of viruses come from? The answers are all over the place in terms of where the different groups, some of them may be descended from cells. We know that several groups of viruses are evolved from plasmids. Where did plasmids come from? Well, that's a good question. Probably the same mush of chemical reactions from when cells first appeared. You have all these different biological entities, cells, viruses, plasmids, viroids, transposable elements. They all do kind of the same subset of things. There's a lot of overlap in terms of their activity. And importantly, there's a lot of overlap in terms of their sequences. You can find homology across all these different groups. So I think if you go back 4 billion years, that's where you get all these different groups kind of specializing in their life cycles. And that's where I don't have any evidence for that directly. That's just me thinking, like, that's what I think explains it. But like, you need to go back and see. And we can't go back 4 billion years and see.
Yeah. So call me email. Evolution doesn't only protect what we should find in biology, but it can also protect what we should not find. Design can't make the second prediction because it would imply that the designer couldn't create the design.
Oh, yeah.
Exactly.
I hadn't thought of that, but yeah, if you're depending on how you characterize the designer, if you're saying, well, we shouldn't see X, then you're imposing limits on what that designer is capable of. Yeah, I hadn't thought of that, but that's an interesting approach.
Yep. Another great question. Can some viruses be genes became autonomous from horizontal gene transfer, for example?
I'm not sure exactly what's going on there, but I think so. You can have genes from viruses that kind of move to their host organism via horizontal gene transfer. We know that happens. You can find those. The best example, I think, are the syncytins in mammals that are involved with developing the placenta. And those are homologous with genes from retroviruses. So a retrovirus inserted its genome into our DNA, and then it turns out if you express that gene during embryonic development, it allows you to suppress the inflammatory response in the uterus, and that promotes long-term gestation. So it's a key step in building placentas. So that's a really, really cool example. And it also goes the other way. We can have entities like plasmids become viruses, and we've actually recently, within the last 5 years or so, I think, we've actually observed this experimentally, where you have plasmids infecting some prokaryotes. They were archaea, which, if there's any archaeobiologists watching, I'm using prokaryote shorthand here. I know archaea aren't really I know, but just go with it. So this was plasmids that infect archaea, or archaea, depending on how you want to say it. And what they found was the plasmids were transmitting from cell to cell in lipid bubbles, which plasmids aren't supposed to do that. Plasmids are not supposed to do that, but they did. They started doing that. So yeah, that's there's a lot of horizontal gene transfer that goes on. It's really
Yep.
important.
And really, horizontal gene transfer and vertical gene transfer, those are very interesting stuff.
Right, and vertical gene transfer for everyone. So for everyone, just be on the same page. Horizontal gene transfer is like from individual to individual. So like a bacterial cell has a plasmid, and then it does conjugation and makes a little tunnel, and then the plasmid replicates and goes into the other bacterial cell, right? That's horizontal transfer. It's individual to individual. Vertical transfer, parent to offspring, right? So it's vertical, generation to generation. So you can have horizontal gene transfer is from thing to thing. You get that across species a lot. Viruses often mediate horizontal gene transfer. And then vertical gene transfer, of course, is just having kids. You're giving your genes to your kids. That's vertical gene transfer. Right? And that's how you move genes between organisms. Those are the two options.
Okay, and Creo, I love these questions. Keep them coming. What is your favorite example of one cellular to multicellular life? I think I can answer that question. Personally, for me, I think there was a paper published in Nature a few years ago where they actually took unicellular life and subjected them to degradation, and they became, over time, obligate multicellular organisms. So that is my personal favorite.
Chlamydomonas.
What was that?
That's a great. It's Chlamydomonas is the green algae, and it's a unicellular little guy, and it zips around. If anybody has if you have just any old cheap microscope, you get a couple drops of pond water, and you put it. You'll probably see some Chlamydomonas in there. It's super common unicellular green algae. And it turns out clumping up is a really good anti-predator defense. And we've known that. They'll clump up. Like you'll have multiple individuals that will express genes that'll say to each other, hey, let's clump up. There's predators around. And what they showed in this experiment, so cool, is that one, they became obligate multicellular. So they stopped doing any unicellular stuff at all. Two is that it was clonal rather than aggregate. So what they would do in nature is aggregate together. So you have individuals that are separate from each other, and they would just clump up almost like a blood clot, right? But what they did experimentally is they would split, but they'd stick together. So they'd make a clump of cells clonally, right? And then they couldn't live separately from each other. They were obligate. So they were true multicellular. So cool. And even when they got rid of the predator, they stayed multicellular. So cool.
Oh, yeah. That is really interesting. So, Gutsick and Erica, I was definitely going to ask this question, but can we get what's your favorite virus? You have a favorite virus?
So I have, my personal virus that I worked on all through grad school was a teensy little single-stranded DNA bacteriophage called Phi-X-174. And so it's always going to be near and dear to my heart because I spent years and years and years transmitting this little thing from culture to culture, doing all sorts of terrible things to it along the way with mutagens and such. So I love Phi-X-174. And fun fact, Phi-X-174, it's a teensy little single-stranded DNA genome that was, it's only about 5.5 kb, so about 5,500 bases, not even base pairs, just bases, because it's single-stranded. It's a tiny little circle of DNA. It was the first DNA genome ever sequenced, was Phi-X-174. And so that's really cool. That being said, I love the giant viruses, the subgroup of the group called the NCLDVs, the nucleocytoplasmic large DNA viruses. A subgroup of that are the giant viruses. You get your Mimi virus, your megavirus, your Marseille virus, your Mama virus, cafeteria virus. There's dozens of them now, and I don't know hardly any of the names. But the biggest one is one that we isolated from Siberian permafrost. And this is a true story, but this is how zombie apocalypse movies start. We isolated a virus from the permafrost. It's the biggest virus we've ever discovered. They called it Pandora virus. And then what did they do? What did they do immediately? They said, I wonder if this virus still works. And wouldn't you know it, it still works. It infects amoeba. It works. So That's a thing that happened for real. And those giant viruses are my favorite because they are larger than some cells. They are more complex. They have larger genomes. They have more genes. Why do these things exist? They have genes from all 3 domains of life. So it's just, yeah, I love those big ones.
Priya with another fantastic question. What are the reasons AGCT/U bases were selected as code? Is it because ultraviolet resistance? How do you think? I love this question. This is a great question. Thank you, Creo.
So there's a paper. I'm gonna find it. I'm gonna find the link to it real fast. You can put it in the side chat on there because it's one of my favorite papers of all time. It's called Confounded Cytosine, and it's just like that. It's got an exclamation point. It's Confounded Cytosine, and it's I forget who I've said this to. But cytosine is like my favorite thing, like my favorite molecule. Like, what is my favorite molecule? Forget virus. What is my favorite molecule? It's cytosine. Cytosine is the most fascinating thing on Earth because it makes no sense. So like cytosine is so dumb because it mutates really fast and it does this specific thing called deamination, right? And when cytosine deaminates, what does it become? It becomes uracil. Now, we know that RNA came before DNA, right? We know that RNA was first, then DNA. That's that confounded cytosine paper. So here's the fun part, and this paper goes into all this. You have your RNA, okay? You've got your cytosine. Your cytosine turns into uracil. Well, how do you know when that mutation happens? Because uracil also belongs there, right? How do you know when that happens? Well, here's an interesting thing. We have a molecule that's more stable, DNA, and that's going to be a better medium for storing your genomic information than RNA, right? But we still have this cytosine to uracil problem. What's the solution? Well, what if we use thymine instead? And I'm personifying this. This is all just based on selection and stability and stuff, right? But what if we use thymine instead? Because now, if you have DNA and you use thymine, anytime you see a uracil, what does that mean? That means that should actually be a cytosine. So in all of our cells, not just us, but every form of cellular life has tons and tons of enzymes that all they do in your cells all day long, 24/7, is they prowl your DNA looking for uracil. And anytime they find it, they cut it out and it gets replaced with cytosine because of this stupid mutation thing. And that's why we use thymine instead of uracil in our DNA. It's because cytosine is stupid. And it's this paper, this confounded cytosine goes into this. And it's such a good read. I love everything about this thing because it gets at the earliest history of biological systems. I'm like, how did this actually happen? And the kind of the subtitle of this is I want to get the wording right. It's Tinkering in the Evolution of DNA. And that's so perfect because it really is just like futzing around with it. It's not, if this was designed, there wouldn't be cytosine in the first place. We'd find something better that doesn't mutate to one of the other things that we use. It's like reading a book where like some fraction of the time, all the A's just spontaneously turn into E's. And it's like, well, that's dumb. And then what do? We found a different way to write E. So when A turned into E, we knew that that's actually supposed to be A. Well, no, that's stupid. Just fix A, right? But we didn't. We came up with this workaround using thymine instead. And it's such a beautiful illustration of how evolutionary processes work. And that's, yeah, I love that.
Yeah. And does Dr. Dan think the premise of the movie The Fly with Jeff Goldblum might be possible? I'm not familiar with that movie.
I'm actually not familiar with that movie. I know the idea of it, that Jeff Goldblum turns into a fly. I'm going to go with no.
Right. And so, yeah, we've been going on for about an hour, a little bit over an hour. So this will be a pretty good place to wrap things up. One final question from Creo. So there may be other bases that work better.
If you were to design a genetic system around 4 nucleobases, you would not pick cytosine. You would find something else that matches with guanine that's not cytosine, and it would be way more stable. Now, There is, I will say though, I'm like ragging all over cytosine here, but I will say that we do see in certain cases selection for higher mutation rates. So there's this balance where it's not, mutations are useful because mutations are your source of variation, right? So it may be the case that early in history, we actually had multiple systems. And the trade-off for being more stable was a lack of adaptability because you didn't have the variation. So it's possible that the system we arrived at wasn't just like, well, it works, so that's what we are now. It actually won in a competition with other systems. We have no idea if that's the case, right? We can't characterize what existed way back then, but it's certainly possible, right? Because we know there are organisms right now, and my little pet virus, Thiox174, is one of them, where they actually suppress DNA repair mechanisms to artificially raise their mutation rate. And the fun thing is with that virus, if you put it in a mutagenic environment, you end up leading to mutations that permit E. coli. Its host is E. coli. It's a phage that infects E. coli. It actively evades the DNA repair that E. coli does. But if you put it in a mutagenic environment, it will actually, you'll select for mutations that permit E. coli to repair it. So you can lower the mutation rate by an order of magnitude by putting it in a mutagenic environment. It's so cool.
Yep. So basically another observable, derivable refutation of genetic entropy.
Yeah, no, really. Yeah, it's literally, it will decrease its own, you will select for a lower mutation rate when you hit it with more mutations. Yeah, yeah. I hadn't thought of that, but yeah, that is a direct refutation because that shouldn't be possible otherwise. Yeah, you're right. Thank you.
Yeah, anyway, thank you guys so much for joining me. This has been so much fun. Dr. Dan, what stuff do you have coming up on your channel?
Oh man, so I have a, let's see, so my next creation myth is going to be on the very common claim that we hear that the new activity, this is like very inside baseball, but it's very specific that we hear all the time, that this new biochemical activity in HIV is that that's actually not new. This tetherin antagonism in a protein called VPU, it's actually not new. It's actually just recovering an ancestral trait. Yeah, that's not true. It is completely new. It's a novel mechanism to do this thing. So I'm going to do that. I just have to like sit down and record it at some point. But that's what the next creation myth is going to be. And then I, heaven help me, I'm going to talk to Danny about a natural selection at some point, I think. I'm going to set that up. Because I'm now about halfway through Fodor's book, and let me tell you, it's not good. And so we're going to talk about that. I think I'm going to subject myself to one more conversation on that. And then I got a few other things that are more up in the air. I got a few people coming on I just need to set a date for. So I'm working on that. And there'll be more details when I have them, but my summer class starts on Tuesday, so we'll see.
Yep. And awesome. So thank you, Dr. Dan, so much for being here. We definitely should do another part 2 of this sometime. I really enjoyed this conversation.
Hey, twist my arm. Make me nerd out about viruses for an hour.
Oh, yeah, definitely. Anyway, thank you guys so much for joining. Have a wonderful rest of your day. And join me on Saturday at 7 p.m. My friend Brenton McGee is going to be here talking about music, taking a break from science, and just talking about music.
Nice.
So if you like classical music, if you like opera, that's the show for you. And that is at 7 p.m. on Saturday. Anyway, thank you guys so much. We are out. Bye-bye, everyone.